Okay, so I'd like to start by introducing Anne Tokiyoda. Anne came to Iolani as a new junior this year, and from her very start, her passion for marine biology has shown through. She took on this new and exciting project developing eDNA for our native opie shrimp, which, like all new projects, required extensive troubleshooting and long hours after school, which she always did with a smile. Okay.
Hi everyone, my name is Anne, and I'm conducting research to detect native opie, an endemic freshwater shrimp, using environmental DNA in freshwater streams on Oahu. I chose to do this research project because I was so curious as to how we can detect species in an environment without ever seeing them. Through my research process, I have affirmed that using an eDNA approach for detecting rare species, such as native opie in Hawaiian streams, is an effective tool that can be utilized to detect species in a stream. So what are native opie, and why have I chosen to commit so much time and energy into these species? Opie is a common word most of you probably know as shrimp. However, besides being used generically, there are actually only two native opie species on Oahu, Opie kalaole and Opie oaha'a. These freshwater shrimp are endemic, and they are especially special because they migrate from the oceans into our streams over the course of their lifetimes. Native opie are culturally and biologically important to Hawaii and are a native decapod crustacean that resides in streams and estuaries all over Hawaii. In old Hawaiian newspapers, native opie were not only used as food sources, but also as bioindicators of weather changes and stream health. Before major infrastructure changes, endemic species like native opie would migrate with the moon phases, but nowadays not much is known about their migration patterns due to the disruption in their lifestyles. The native opie populations are becoming scarcer due to invasive species and habitat loss and degradation. Because opie are an endemic species as well as an anandromous decapod crustacean, which means they live in both saltwater and freshwater, they have greater challenges to survival. Native opie oaha'a, which is pictured on the left side of the slides, is the more commonly found species of the two, as they prefer low flow streams and low altitudes. Yet, through manual monitoring, population detection has become scarcer with time, making them a cryptic species in recent years. Native opie kalaole, the rare species on the right, are actually marked as a threatened species. So they prefer extremely high altitudes and high flow streams and are climbers and climb up hundreds of feet high waterfalls. Native opie kalaole have never been manually detected through surveys conducted by Paepae of Waikolu, which highlights the rarity of these species. So we know through all of history that traditional manual monitoring works. In fact, the manual monitoring program here at Iolani, Paepae of Waikolu, is one that has been fundamentally necessary. Not only does the program conduct community-based mass stream surveys, they also pull out all the invasives they catch out of the water to prevent further invasive takeover. However, there are limitations, such as time costliness, need for manpower, and possible taxonomic error. For example, this native opie oaha'a was caught in a Tahitian prawn net. Can you easily tell the difference between the two species without some sort of expertise in taxonomically identifying them? So on the right side is the native opie oaha'a, and then on the left side is the Tahitian prawn, which is a rare invasive species. This is when eDNA can play a crucial role in detecting species presence to fill in the gaps manual monitoring may miss. So Paepae of Waikolu does lengthy and accurate stream surveys, but has minimally detected cryptic species like native opie. So can eDNA detect cryptic species due to its more sensitive monitoring, or add to stream surveys by detecting what we can't see? Thus, I've chosen to use eDNA as a method to detect the native opie. Using the eDNA method is amazing because it reveals the presence of otherwise cryptic species from just sampling the water, in addition to the mass stream surveys to see if eDNA can catch something our eyes can't. Therefore, the purpose of my research was to firstly optimize my decapod PCR primer and standardize procedures to better amplify DNA in order to successfully and accurately detect decapod crustaceans, and secondly, to detect native opie using an eDNA approach. So we were able to detect decapod crustaceans after a lot of troubleshooting. So I collected waters from 4 streams known to have observed native opie, which is Marketplace, Kaimuki Stream, Waiihi Stream, and Waikeakua Stream. We collected 50 to 500 milliliters of water and ran it through 3 different filtering methods: centrifuge, Sterivex filter, and 0.2 micron filter. We found that centrifuging the stream water, then immediately processing it and extracting the DNA directly after was the most effective and had the greatest DNA yield. At this point, we are still in the environmental sampling stage. I then extracted the DNA using a DNeasy kit and Monarch kit and didn't find a difference between the kits aside from the price point. The DNA extraction kits extract all the DNA out of the water and essentially take the concentrated DNA in the water through breaking down the proteins in the DNA strands. After the extraction, I used the decapod primer's MyDeca to PCR amplify the decapods from my water sample. This is a group-specific primer and is part of the PCR process. PCR amplifications using group-specific primers only amplifies decapod DNA, so it filters out the rest of the DNA in the water like pigs, fish, humans, more. I had a lot of trouble with the size of the PCR reaction in primers, as well as finding the best temperature range for the touchdown PCR. This is because too high or too low temperatures can either heat up the DNA too much or not enough so that the DNA isn't released. This PCR reaction separates the decapods from all the other animals in the stream, and the touchdown PCR, or the heating of the DNA following the PCR reaction, brings the DNA to the perfect temperature so that it contains the high sealed DNA following the extraction. After optimization, I can confidently say that if decapods are present in the sampled water in a high enough concentration, their presence will be confirmed through an agarose gel, which indicates DNA presence. Once our PCR reaction was finally consistently successful, I continued on with the rest of the standardized procedures with eDNA, which includes high throughput sequencing, bioinformatics processing, and species identification. To simplify these words, it means that I took the PCR product and sequenced it to find out what was in the water. I sequenced the PCR product using the native barcode kit on a Monayon sequencer, and I blasted the DNA, which stands for Basic Local Alignment Search Tool, or BLAST. This finds regions of local similarity between protein or nucleotide sequences based on statistical significance of the matches. Essentially, blasting the DNA is just like using Shazam for DNA. I then use those clusters from that blast to find specific species. I know the bioinformatics processing is reliable, as in my settings, I had any match greater than 95% classified as a read or a certain species, leading to accurate species identification. This process is essentially matching 16S whole genomes to the animals present in the DNA and spitting out the species nicely for us to see. So I used a data bank called GenBank, or NCBI GenBank, which has almost all animal species accession numbers, which is the whole unique genome for each species. I comprised a small database with both invasive and native species on Oahu and ran my results against the curated database as well as the whole GenBank database. I found that using a curated database is most optimal for when you're looking for a specific species like I was, but if you're doing a general stream survey, running it against the whole GenBank database will provide more results of various species. So I wanted to mention the troubleshooting because it was a huge part of my project. From hard to locate contamination, as you can see by the false negative on my PCR confirmation gel run, DNA extraction altercations, PCR touchdown fixing, all the way back to filtering methods, I found out a lot about the methodology involved in eDNA. And I've realized that while there are many studies out there studying eDNA and supporting the use of it in day-to-day monitoring, there's still so much we don't know about optimizing eDNA so that we come out with clean-cut results every time. Like manual monitoring, eDNA has other uncontrollable limitations. Unpredictable weather, the temperature of the water leading to quicker degradation of DNA, and many more make it all the more important that any information on results of eDNA extended to natural environments are shared. So I hope to share my findings through publishing to inform other eDNA researchers about my experience with optimizing the eDNA process, especially as there aren't many studies concerning decapod detection. Referring back, I have results from 4 streams in which the eDNA survey was able to detect decapods. These were the Manoa Marketplace stream, Kaimuki stream, Waiheke stream, and Waiakea stream. So these are the results from the sequencing. This is Manoa Marketplace, and we were able to align the manual monitoring and environmental DNA detection of these decapods in most streams. However, as you can see in Manoa Marketplace, I was able to detect prairie shrimp with my eDNA assay, whereas the manual monitoring could not. This is Kaimuki High School, and we were able to detect cherry shrimp and Tahitian prawn, which are both invasives, where the manual monitoring program couldn't. And cherry shrimp are really small, so it could be that they're hard to manually monitor, and Tahitian prawn are really rare invasives in Kaimuki. So it's actually really interesting that we were able to detect it with an eDNA assay. However, Kaimuki was the only stream which I didn't sample on the same day as the Paepae o Waikolu program. So they were able to detect native Opai or Ha'a where I wasn't. And then this is Waiheke stream. So I was able to detect Tahitian prawn where the manual survey wasn't. and the same for Waiakea colostrum. So the eDNA survey was able to detect decapods not collected during the manual survey, indicating additional invasive decapod diversity in Manoa Marketplace, Kaimuki, Waiheke, and Waiakea colostrums. And all streams have detected native opie in the past, but in all stream sampling, except for Kaimuki streams, we sampled the same days as the program. So although the eDNA analysis added a lot more information to the manual surveys about what species are present, the eDNA analysis was not able to detect native opie. And if opie are detected, it can help ecologists in Hawaii to more accurately gauge the biodiversity on Oahu, especially regarding these native species. The ability to detect Tahitian prawn, a rare invasive in these streams, shows how eDNA is able to detect cryptic decapods. And if a greater population of native opie were present, it is likely we could detect them with an eDNA assay as well. If native opie aren't detected in the future, it may be that eDNA is not the most effective detection method, but if they are minimally detected, it's a sign for conservationists and the general public to implement efforts into preserving this crucial endemic species. Through working with clean streams with significant pollution, as well as modifying modern obstacles of infrastructure for habitat restoration. I learned so much this year from literally knowing nothing about eDNA to being able to do a PCR reaction with my eyes closed. I have truly learned about not only DNA, but about the research process through my mentors. And the advice I have for students is definitely persevere, because I faced a lot of issues, especially with contamination, and I felt like nothing was working and my research project would never work. But with research, you have to read into a bunch of studies, you have to understand the process you're doing, and through that, you will succeed. And I also say that you should definitely have mentors who are just as excited about your research as you are, because I couldn't have done it without their expertise or unconditional support. Speaking of, I'd like to thank my mentors. Mrs. Kobayashi, you have become my biggest role model and supporter. You have put so much time and effort into me in and out of the lab, and I cannot emphasize how much you have inspired me to continue my research, even when I was frustrated and tired and wanted to quit because nothing was working. Under your wing, you have built my confidence into becoming a scientist myself, and I want to thank you for your unconditional support and kindness. Mr. Hill's not here, but I would like to thank him for teaching me how to make a database and to use fancy words like R language, Dakota Pro, and GitHub when I still can't even touch type, but also for his patience beyond extremes and all the hours and hours of work behind the scenes he has done to make my project successful. Also, he pushed me a lot to do everything despite my computer skills, and I sure wasn't grateful for it back then, but now that I can make colored pie charts, I have him to thank. Dr. Chan, coming to Iolani this year from a public school that didn't have research opportunities like here, I distinctly remember when you suggested eDNA as a research project, and I thought you sounded like that scientist from Jurassic World. Amplifying DNA sounded so daunting, yet you made me feel so welcomed and have taught me so much since then. Thank you for introducing me to the world of eDNA and science, and also giving me advice on my career beyond high school. And special shout out to Mr. Corey Yap and Ms. Andy Sharuk, who work in the Iolani Community Science Program and conduct manual survey streams in Paipai o Waikolu. I want to thank them for teaching me about the significance of native opie, giving me true advice and supporting the growth of me as a scientist. Also, thank you to Mr. Yap for coming out when Dr. Chin and I were sampling the stream, just to stand in the pouring rain and watch as I try not to drop the sample bottle. And thank you all for listening to my project.